Good morning. Welcome back to Open Med School. This is part of Physiology Complete. Today I am going to discuss with you the competency-based physiology PY 1.5 transport across cell membranes. As we know, in order to maintain the integrity of the cell, transport of molecules is essential. So let us think about the mechanisms involved in this transport. Basically, it can be divided into two. One is the active transport, second is the passive transport. Active means it is energy dependent. There is usage of energy basically in the form of ATP. And other is passive. Passive means there is no energy use. So today we'll be discussing about the passive transfer of or transport across membranes. So let us take a cell. All of us know cell is a, so if you draw, I request all of you to take a paper and draw the structure of cell. We know that the cell membrane, if you take a part of the cell membrane, this will look like this. By lipid layer. This part is the lipid. So it is the what is called the bilipid. Only lipid soluble materials can pass through. For example, if we take oxygen, oxygen can pass through. Carbon dioxide is inside the cell. Inside cell there is carbon dioxide that can diffuse out. The classical example is that what we see in the lungs. This type of diffusion is called simple diffusion. This is an example of a simple diffusion. Hope this point is clear to you. But again we take a cell. Particles which are big, for example sugar, or charged particles like sodium, potassium, they are charged and they can't pass through it get back. So then they cannot pass through the membranes. In order to transfer such materials, we require certain, or we require facilitation. In the cell, there are protein molecules which act as channels for passage materials. For example, the sodium can pass through, they are called the sodium channels. When the potassium, it is for the potassium, they are called the potassium channels. So they are specific channels are there for the passage of ions. So that is an example of a facilitated diffusion. So we have discussed about the simple diffusion, where the only small particles can go, and in a facilitated diffusion, charged particles and larger particles like glucose can pass through. And all of us know all the cells require glucose. Glucose is a large molecule and it cannot pass through the cell membrane. So there are channels called glucose transporters or the blood. Glucose transporter. Glucose transporters are a family of protein channels. In different cells, there are different classes are there. We'll study this in detail later. These channels gets activated when the hormone insulin, all of you know hormones, Here it is insulin. Insulin is secreted by the beta cells of pancreas. There are receptors on the surface of the cell. They are called the insulin receptors. So once the insulin gets attached to it, there are signaling pathways are there, and this glucose channels are there inside the cell. So this once the insulin attaches, this will move towards the surface of the cell and get attached to the cell membrane and form channels. The glucose will enter the cell. Here again, remember that there is no energy is required. This is facilitated by a hormone, insulin. Such channels are called ligand-gated. So only once the insulin gets attached, these channels get activated. So this is the example of a ligand-gated. There are different other types are there. For example, there are acetylcholine or other neurotransmitters are getting attached. Activation, of course, I was just said about glucose. We have said about the ion channels. Let us look a little more detail about the ion channels. So again, this is a cell. So these are, we have mentioned about the channel. This is an example of a channel. Let us take, it is the potassium channel. This channel is open always, and all of you know the inside of the cell contains more of potassium and outside contains more of sodium. So there is a concentration gradient for the potassium. That is inside the cell, excess of potassium. So what happens? Potassium will leak outside. So they are called the leaky potassium channels. Ion channel, leaky potassium channel. Leaky means depending upon the concentration gradient, the substance will move. So diffusion is a process, not energy requiring. Wherever concentration is more, it will move out. In simple diffusion and in facilitated diffusion like ion channels or ligand-gated channel, the diffusion is from a higher concentration to a lower concentration. There are certain other channels. So example is the sodium channel. These sodium channels will get activated only when a gate opens. So such channels are called gated channels. So the sodium channel has got gate. Gates are simple polypeptides. They are again peptides. So certain situations only the gate opens. Some of the gates open when there is change in the voltage. Such channels are called voltage gated channels. So we have mentioned about the leaky channels. Classical example is a potassium channel. Then we have got voltage gated channels. These channels respond only when the voltage changes. We'll study in detail about when you study about the membrane potential. You know that the interior of the cell is more negative and outside is positive. Many cells it is approximately minus 70 to minus 90. By some reason, when the membrane potential changes to minus 60, that is inside becomes less negative. What happens? These channels opens. So that is an example of a voltage gated channel. Hope this point is clear. Some other channels are responding to mechanical causes. They are called mechanically operated. A pressure change can produce a opening of the channel. We have already said about the ligand-gated channels. So all these things are examples of facilitated diffusion. One more example we can study. This example of a facilitated diffusion is in the neurotransmission. You remember this is the thing at the terminal of the neuron. In the terminal of the neuron, the neurotransmitters are situated in synaptic vesicles. They are released by a process called exocytosis. We'll study it later. So this occurs when calcium enters a cell, and we will think about how these synaptic vesicles store the neurotransmitter. Some of the neurotransmitters are stored. One is the glutamine, another is acetylcholine. So many neurotransmitters are there. So they are synthesized inside the neurons by a carrier mediated. They enter the synaptic vesicles. So entry of neurotransmitter and storing it in the synaptic vesicle is again another example of facilitated diffusion. So we have discussed about simple diffusion, example is the oxygen carbon dioxide. We had discussed about the facilitated diffusion, example glucose transport, ion channels which are divided into leaky channels, voltage gated, mechanical, ligand. And examples also we have mentioned. Now we go to one more passive process, that is osmosis. So far we have discussed the diffusion divided into simple and facilitated. Now we are going to the osmosis. So what is osmosis? Suppose there is a semi-permeable membrane is here, and it contains a solution contains lot of solids. For example, sodium chloride, and here is the other side it is only water. This is a concentrated solution, and water molecules will try to enter. This is what is called osmosis. Regarding osmotic pressure and other things we will study later. This is only just introduction to osmosis. So a solution containing large quantity of solutes are called hypertonic solution. With the low quantity, it is called hypotonic solution. The concentration is same as that of a cell. It is called isotonic solution. So 3 terms you should remember. Now come to the clinical relevance of this tonicity of fluid. Let us take plasma. In the plasma there are RBCs. If the concentration of the solution in which the plasma, this is the blood vessel, and if the concentration of solutes are more example, more of sodium chloride, what will happen? The water will from the RBC enter the plasma, and what will happen? The RBCs will become crenated. The shape of RBC is crenated. Suppose it is a hypertonic. Comparing the RBC, if the solution is hypertonic, what will happen? Fluid will enter the RBC. So what will happen? The RBC will get enlarged. It will burst out. So that is the importance of, okay. And in relation to the osmosis, again remember, for water molecules, water can go without any particular channels, that is by osmosis. But there are certain channels called aquaporins. Again, there are different types of aquaporins. Aquaporins are channels which allow the movement of water. Again, we'll study the clinical relevance later. So in summary, today we have discussed about the passive simple diffusion. Example is the oxygen, carbon dioxide. We have discussed about osmosis, water is moving. We have discussed about facilitated diffusion. Facilitated diffusion, you have said about the ion channels, the leaky ion channels, and the gated channels. Gated channels are voltage gated, ligand gated, mechanically operated. Hope this was useful to you, and we will discuss active transport in the next session.